Hello again, and welcome to Lab 2. My name is Ms. Copay, and today we're working on cell permeability. So this is going to actually connect back to our biomolecules from last unit into Unit 2 on cell structure. And what we're going to be looking at today is a semi-permeable membrane. So remember how our cell membrane allows some things to pass through and other things not to pass through. The kinds of things that pass through our cells easily are things like gases and water molecules. Things that don't pass through our cells easily are big molecules and things that are charged molecules like sugar. And today we're going to be looking at something like starch. So those molecules are not going to be able to pass through. Small molecules like our gases, even things like small ions like iodine here, they may be able to pass through our membranes. So we're going to be looking at those properties of what can pass through and what cannot pass through a selectively permeable membrane. In our lab today, we're going to be using dialysis tubing. And if you've ever heard about dialysis, somebody who is suffering with kidney failure or may go through dialysis treatment. And that's because the job of our kidneys is to actually filter our blood and remove all of the impurities so that our blood isn't filled with waste. And so that's the job of our kidneys. And people who have problems with their kidneys, the blood has to be filtered outside of the body using a process of dialysis. And so today we're going to use that kind of dialysis for understanding what semi-permeable membranes do and how our cells work. So in your little packets here, in your kit, you'll find dialysis tubing. You're also going to need starch, the bottle of starch, and your iodine. Additionally, you'll find in your packet that has string and balloons, those materials as well in this packet here, are rubber bands. And you need to have the rubber band so that your dialysis tubing doesn't leak. So you'll find those components here. You're also going to need a little small container as well as your funnel. Okay? So in our lab today, we're going to be looking at these materials. Okay, so here's our dialysis tubing. Here's our bottle of starch, the iodine solution. You'll need a beaker and some water, your rubber bands, and your funnel. Okay. You won't need any other materials outside of those materials in the kit. However, you do need a cup. So grab a cup, and you can use the one in the kit. It works fine, but you may need a second cup. So you might have that around. All right, so to start our lab off with, first of all, I'm going to refer you to the video that's on Buzz. There's actually a demo on setting up this lab. And so you can refer to that video here for walking through the steps of the lab. Okay, key one, the first tip for doing this lab is you have to pre-soak your dialysis tubing. Okay, the dialysis tubing will not work right if it's not hydrated. And so we're going to take it out of our package and we're going to transfer it into a beaker of water. Okay, and so you're going to have your dialysis tubing here. And it's pretty long. We don't need that much. We need our bag of dialysis tubing to stick in our water. So you really only need to cut off a section of your dialysis tubing. Maybe about 4 inches is enough usually to fill it up because it needs to fit inside of our beaker of water. So we're going to hydrate it properly in our beaker. All right? And so give it 10 minutes to do that. While you're waiting, make sure you have your starch and your iodine here. So inside of our bag, we're going to fill our bag with the starch. So once it's hydrated properly, you're going to pull it out of your container and you're going to find that you're going to tie off one end of your tubing by twisting it, and you can even knot it. You don't have to use your rubber band for one end. I knot one end of it down at the bottom. Give it a little snug pull. Okay. And that's going to create a container for our starch to go into. Okay, now just by twisting it between my fingers, I can get my starch inside of here. Okay, so I'm going to use a funnel to make and use my fingers to make an opening at the top end of our dialysis tubing. And that point of that funnel is going to be set right into that dialysis tubing. Okay, so I can shove it down into my dialysis tubing through that opening. And I'm going to put it in this other beaker here so it's easier for me to handle. And I'm going to fill up the contents of this tubing with my starch solution. Set this here And we want to make sure that we don't overfill it. Okay, so you can see it's filling up my starch container. Okay. All right, and that's plenty in my baggie in here. Okay. And it's a little loose, it's not jam-packed. And now I'm going to use my rubber band to close off and seal it. And you want to make sure that your bag is not leaking. All right, so you're going to set this up here. So I have my baggie with starch contained inside of here, the rubber band tied off and the other end knotted, and it's not leaking. Make sure you rinse this bag off so that you get any starch that may have spilled off the sides, because we don't want that to interfere in our beaker. We're going to see that when we add water to this, and we add water covering this beaker here, we're going to add iodine to the outside of it. Okay, so we're going to pour a little bit of iodine in, and that iodine is a brown color, and so you can see your brown color in your solution here. All right? And what we're going to observe here is what happens to that starch. Is that starch able to leave the bag? Is the iodine able to get into the bag? And I'm going to show you a reaction of what happens when starch comes in contact with iodine. So I'm going to use a dropper here, and I'm just going to demo this on a slide so you can see it. If I put just a drop of starch here on my slide, okay, and I take another dropper that isn't contaminated, and I add iodine to it, so this is a separate clean dropper, and you don't have to do this, I'm just showing you what this looks like. So you can see my starch is clear, my iodine is brown, and when they come together, they turn purple. And so hopefully you can see that, and you can see the color difference. Okay, and so what we're looking for is observing which of these solutions here turn color. Does the bag turn color, or does the water turn color? Okay. And we're going to pull it out, and voila! Our bag has turned color. Hopefully your bag will. And hopefully your container of iodine doesn't turn colors. You can see actually when you hold it up into the light that it has a purple hue to it. Okay, so that's your lab. So after you've completed your lab, you're going to refer back to your lab template for doing your write-up, and that's your purpose, your hypothesis, answering the questions from your lab manual, so your lab manual questions that are on page 12. And then complete your conclusion, and don't forget to submit pictures from your work along the way. Good luck with that, and please contact me if you have any questions.